orchids. The diversity of orchids is vast. There are pink ones, yellow ones, red ones, green ones. There are big ones, little ones, ones that look like spiders, ones that look like bees, ones named after vampires, ones named after Kew's Director of Science, Alex Antonelli. In fact, let's say you learn to write at the age of 4. Then, say we asked you to start writing down the name of every single orchid species known to science. Those names can get pretty complicated, so let's make things a bit more manageable. 1 species a day, that's just 2 words every single day. A genus and a species name.
How long do you think it would be before you had them all written down?
1 year? 2? 3? In actual fact, before you would finish your list, you would no longer be a child. You would have finished school. You might have gotten a job or gone to university. You will have made friends, lost friends. You might have fallen in love, fallen out of love, got married, got divorced. Maybe you retired, moved to the south of France, to the coast. Your children are all grown up now. They have jobs and lives of their own. Maybe they even have children too.
By the time you finish your list, you would be only 5 months shy of your 81st birthday. The point is, there are a lot of orchids on planet Earth. You might recognise this orchid, a moth orchid or Phalaenopsis. You may have even kept one as a houseplant. I know I have. Is yours still alive? I think I overwatered mine. I tried the ice cube thing, it just didn't work. But with orchids like this available in so many shops as houseplants, you might be fooled into thinking that orchids aren't a priority for conservationists. But orchids are in danger.
We have about 56% of all non-orchid diversity under threat of extinction.
This is Oscar Perez-Escobar. He's a Kew scientist and orchid fanatic.
And so how many years now have you been studying orchids?
Ay, caramba. Sorry.
Oscar's research has been focused on building an orchid tree of life, examining the DNA of orchid groups to understand how they're related to each other and how they evolved. He's travelled all over the world looking for rare and endangered orchid species.
So with over 30,000 species of orchids, some people might think, it doesn't really matter if we lose 1 or 2 here and there, but why is it important that we protect these species?
Let's say that if we start by thinking of us as humans, orchids are a source of chemical compounds that are useful to humankind. For example, the orchid there that you see there, that one produces a compound that can cause apoptosis, which is program the cell death in cancer cells. Of the 30,000 species that are known to humans, only 400 have been investigated for the useful chemical compounds. As a matter of fact, there are some compounds which are unique to the orchid family. So there is so much to discover, and it's diversity that we are losing without even studying it.
So we're really just kind of scratching the surface on some of these. There could be incredible compounds out there that we just don't even know about yet.
Yes, and if you think beyond what is useful to us, there is also the fact that all of this diversity that has been evolving for the last 80 million years is just being lost. All of these processes, all these interactions with pollinators, with fungi, with other plants are just disappearing without us really understanding it.
A wild ecosystem is in balance. Imagine a giant tree. It's covered in aphids that are feeding off the leaves and nectar the tree produces. Those aphids are eaten by beetles scurrying around the tree. In turn, those beetles are eaten by small birds, and those small birds are predated on by something larger, let's say a bird of prey. Just like this tower here, it's all in balance. The tower in front of me here relies on all of the bricks supporting one another to stay strong and upright. Similarly, our ecosystem relies on all the constituent parts being in balance to stay steady. So what happens if one of those pieces disappears? Let's remove the beetle from the ecosystem. Now there's no beetle around, those small birds won't have anything to feed on. Their population's going to drop, they may even disappear entirely. And with no small birds around, the bird of prey isn't going to have much to eat. It might leave the area, it may even go completely extinct. And with the beetle gone, there's nothing feeding on the aphids. Their population's going to boom, and they're going to be far more damaging to the tree they live on. They may even cause an impact to its health or kill it entirely. As more and more falls from our ecosystem, it starts to get shakier and shakier. We can't know what the final brick is going to be, but eventually one's removed and the whole thing falls to pieces. Orchids are very rarely keystone species. They're not going to be the crucial brick that brings an entire ecosystem down, but they might be the indicators of the first wobbles in the tower.
We sometimes refer to orchids as the canary in the coal mines, and this is because orchids depend on many different organisms, right? They have a strong connection with fungi, with pollinators, right? And when the health of an ecosystem starts to decline because of climate change, deforestation, so orchids disappear because they depend on these organisms, right? So when you go to a forest and you find it devoid of orchid diversity, it might mean that this forest or this ecosystem has been affected by some sort of like disturbance factor.
It's clear that orchids are worth protecting. They're beautiful, they're useful, and they're key indicators in the health of an ecosystem. So what can we do to help?
And because they are very specific to their microclimate where they're growing, any disturbance can actually threaten their survival.
This is Bala Kompalli. She cares for the orchids in the tropical nursery here at Kew Gardens. And when we say she cares for the orchids, we mean she really cares.
For me, every orchid is very important because they are outside their habitat and they are under our care.
What are some of the challenges in protecting and reintroducing orchids into the wild?
The challenges are, first, they have a very minute seed. So the seed cannot be grown like any other normal seed sown in the soil. They need special conditions, then they can be only grown in the lab.
This is the stage where you got these seedlings, so you leave them to grow for another 6 months or something. Then they produce really good shoot system. You just leave it, actually almost like nature takes its course, but inside a container.
Sarasan is a Kew scientist. He leads the orchid propagation team, hard at work deep underground in Kew's labs.
If we compare an orchid seed with a coconut, the coconut we eat is the food material for that developing embryo.
Yeah.
So imagine there is no food material for an orchid seed. It is so tiny and microscopic. None at all. There must be some external help. So the mycorrhiza provide that.
Mycorrhiza, put simply, is a fungus that grows with the roots of plants that allows them to access the nutrients available in the soil.
Although a connection with mycorrhiza is vital for the germination of orchid seeds in the wild, orchids often only have a relationship with a small number of mycorrhiza types. If the right mycorrhiza is not present in the soil, an orchid seed will not be able to germinate.
In an experimental setup, what we do is we collect 2 grams each from several locations around the orchid, and then we clump it all together, and then we take a representative sample from that.
Yeah.
So if you want to take 100 samples, you can put all together and then take 2 grams. That means the diversity you get will be very, very high.
How many species of fungi could there be in 2 grams of soil?
A rough estimate, the last 4 studies which I have done, from 2 grams of one sample, I would say around 500 plus fungi.
500?
Yeah, and of those, nearly 20% of them are mycorrhizal. But quite a lot of them are not mycorrhizal.
If you think dating is hard, just try being an orchid seed. There are so many different types of fungi in the soil that finding a compatible mycorrhizal partner can be a huge undertaking. But there is another way. In these jars, Sarasan and his team are able to grow orchid seeds in an agar solution that provides the seed with the energy source it needs to germinate successfully.
The beauty is, in vitro, you can make it faster.
Speed it up.
Whether it is outside, it is autumn, winter or summer or spring, whatever it is, we give them the right conditions, the temperature, the lighting, everything. That makes the seed germinate faster.
It's always perfect conditions.
Always perfect conditions.
In the lab, you don't need the compatible mycorrhiza to get that orchid started. Just a solution full of all the nutrients it needs to germinate. But what are the downsides to starting our orchids off this way? These are all sealed up, these jars in here. So how are these plants able to grow if they're all sealed in that contained unit?
In some cases, we keep the jars for more than a year without opening it. But there is a downside to that. The humidity inside is quite high. So these plants are quite tender. So if we look at a wild plant or plant we are growing in our windowsill or wherever, it has got a protective layer called a cuticle. If we take these plants out, leave it for 5 minutes, they are dried like anything.
Just can't hold on to the water.
Exactly. So these jars are not suitable for growing them if you are planning to plant them in a pot or outside back in the wild or so. So you need to grow them in a in a container where there is air exchange. That gaseous exchange help the plants to develop the cuticle and slowly, become hardy enough to survive.
Slipper orchids are quite incredible. Can you talk a little bit about the slipper orchids?
The name slipper orchid comes from its specific structure of the flower. For example, this one is a slipper orchid called Paphiopedilum insigne, and you can see it's got a lady slipper-like lip. So this is a modification of this flower, and you see in all slipper orchids this kind of structure, whether it is a cypripedium or Paphiopedilum or Phragmipedium or Mexipedium.
So they've all got one of these lips
Yes.
on them,
Yeah.
but they'll just look different in the different types of orchid.
Yeah, because orchids have 3 sepals and 3 petals, and out of 3 petals, one of the petal is always modified into a special structure that's called labellum or lip. So in this case, the lip has become like a slipper.
And does that serve any function? Is there a reason it has that?
Yeah, it is like a little pitcher if you see closely, and then it traps insect when it comes to visit the flower because they are very deceptive. These orchids will mimic either luring the insects in the form of aphid-like structures or a scent, or mimicking like a rotten meat to attract flies. So each orchid has specific deceptive function. So in this case, the plant attracts the insect, the flower attracts the insect for the pollination. When the insect visits the flower, it slips and falls into this trap. And it has to find through only one way to escape the flower, and then it catches its pollinia on its back.
Do we have any slipper orchids here in the UK?
Yes, we have. Cypripedium calceolus is one example. It is really rare and now it's critically endangered. So at Kew, we are doing a conservation program for them, and we have also distributed them to various wildlife trusts who helped to develop this project.
How has it become quite so rare in the UK? How did it get to that point?
In the Victorian times and maybe before that as well, people are always looking for very charismatic plants. And in the UK, there may not be a better plant than lady slipper. It has got beautiful flowers, and the people wanted to have these plants in their back garden. There is only one plant left in the wild. It is in a secret location. It is just to protect it from people who are very passionate about seeing these plants, but at the same time we got a duty to protect the plant from collecting seeds or collecting material.
So Kew here in the in vitro lab, we're growing these seedlings. Have we started making any efforts to try and reintroduce these into the wild?
Yeah, over the last 30 years, I think every year we used to send nearly 1,000 plants. I'm talking about early stages seedlings here. Then once they are hardy enough, they put them back to the wild. And so from seedling stage to get them flower, it may take nearly 8 to 10 years.
8 to 10 years.
Yes.
So it really is you have to be very patient
Absolutely.
to see if it's been successful.
Absolutely.
So you say we've been doing this for about 30 years. How are the results looking at the moment in our work?
Last year, we have got this great news saying that there's a new seedling, a new plant. So that gave a new sort of direction to the research side, mainly because if a seedling to be found, there must be mycorrhiza in soil. Without that wouldn't happen. So our research now is to see whether we can identify the mycorrhiza from soil through growing seedlings in that soil, rather than growing these seedlings in different types of orchid mixes. So if these seedlings can recruit the mycorrhiza, then we will have a bit more understanding about what kind of mycorrhiza is there, which are the ones these seedlings recruit, and if we can get those mycorrhiza and then grow with the seeds, we may be able to find out if there is any mycorrhiza which is compatible.
It sounds like it's a real collaboration between botanists and mycologists as well, because you're having to identify both the fungal elements and the plant elements.
We are not talking about just plants here.
Yeah.
So we are talking about fungi which we never come across before, and we don't know their identity. You may have 1,000 isolates, they all look the same, and then you are trying to pick one from that group.
It must be a bit overwhelming sometimes. You're looking for an answer that's not even been discovered yet, that no one's named yet. So you're trying to find this thing that no one knows where it is.
Absolutely, absolutely. There are, in the UK, even there is only one plant, the availability of mycorrhiza for us to research is practically, no, we can't do that. So the method we are now using is a completely opposite. Rather than collecting roots, we are using the collection of seedlings we got in our collection, in vitro collection, to find the mycorrhiza which is in soil.
It's kind of turning these environments into the lab itself, isn't it? You're testing them out in the wild to find these key mycorrhiza.
Absolutely.
The work that Sarasan and his team are doing here has real potential to save orchids out in the world. The success with the slipper orchid is proof enough of that already. But that's one species out of 30,000. It's a long road ahead to protect all of these orchid species. What is the first step we can do to help protect all of these 30,000 species of orchids?
And one essential thing is actually understanding the diversity we have, because you cannot really protect what you don't know you have.
Of course.
So that's why we in Kew go, and in cooperation with our international partners, we do field work. We characterize the flora, in my case, orchids that grow in specific countries. That is the first step towards protecting something. We released in 2024 a second version of the Tree of Life, where we attain a sampling of 40% of all known major orchid groups, what we call genera or genus. We hope to release this year a new version, which will attain hopefully 85%.
Amazing.
Yes, so we are combining all of the sampling that we are producing here in-house and in cooperation with our partners, but also combining data that is available in the internet, sequence data.
Orchids have adapted to fill almost every single environment on the planet. But with this success comes risk. Because they're so adapted to so many different environments, one slight change in that ecosystem and they can disappear. They're at risk. But that's why our researchers are working so hard to try and protect them. We're naming new species so we can understand how they're at risk. We're returning them from being at the brink of extinction. And finally, we're even putting them back into the wild so they can take part in that ecosystem again. These 30,000 species have a much brighter future than my Phalaenopsis.